The overall goal of this procedure is to use a pH-sensitive GFP variant to study the spatio-temporal dynamics of axon guidance receptor trafficking at the cell surface. This is accomplished by first tagging the receptor of interest with the pHluorin sequence using an appropriate cloning strategy. The second step of the procedure is to characterize the pH sensitivity of the pHluorin-tagged receptor transfected in vitro. The third step is to electroporate the pHluorin-tagged receptor construct in ovo to achieve expression in the spinal cord. The final step is to section off the electroporated embryos at the desired stage. Ultimately, results can show changes in receptor trafficking at the cell surface in both spatial and temporal domains through immunofluorescence microscopy.
The advantage of this technique over existing methods such as the use of GFP-tagged protein constructs is that the pH sensitivity of pHluorin-tagged receptors allows the detection of spatio-temporal changes of the receptor dynamics at the cell surface, both in vitro and in vivo.
This method can help answer key questions in the axon guidance field. Indeed, it can help characterizing the spatio-temporal regulation of a cell surface distribution of an axon guidance receptor. And we know that this regulation is critical to set the sensitivity of a growth cone to a particular cue.
This method can provide insights into the field of axon guidance within the chick embryo model, but it can also be applied to other systems, such as mouse brain slices, maintained in culture in combination with